All right, if you're ready for a journal club, let's do a journal club. The title of this paper is Integrative Molecular Structure Elucidation. They found structures. And construction of an extended metabolic pathway. They reconstructed metabolism associated with an ancient immune response in COVID-19 patients. And of course, we are really interested in the ancient immune response. This is an antiviral response, and we've been focusing on antibacterial, but I don't want you to forget about antiviral, and this is a chance to talk about it. In fact, this is specifically an antiviral response in COVID-19 patients, and beyond that, it's a response that can be detected in the urine, which means we have a diagnostic test to tell how someone's immune system is doing by looking at the urine, which we usually throw away. Very cool. What I find really amazing about this, so the innate immune response is this protein called viperin. And what viperin does is the same thing that Gertrude Elion won a Nobel Prize for. And in fact, it's kind of cool because we are now in mid-January. Very soon it will be Gertrude Elion's birthday. And so she was born in 1918, and she died in 1999. And in her long life, she was able to win the Nobel Prize in 1988 for making new drugs. And her strategy for making new drugs, really brilliant and really cool. Basically, she made nucleotides, and then she modified them. You know, nucleotides are normally A, C, T, and G, and they have specific chemical structures. Well, they're used in all sorts of processes in the cell. So what if we made weird nucleotides and then tested them on different diseases that involved nucleotides, whether in making DNA or in signaling, because like ATP is involved, that's a nucleotide. Cyclic AMP, that's a nucleotide. So these things, they didn't have to know really what was going on. They just tried a lot of different structures. Elion used organic chemistry to make lots of these variants. And so you see these molecules. If you know what nucleotides look like, these should look kind of like nucleotides, but messed up. Because what these will do is they will bind in the same site as nucleotides, but they will mess up that binding. And sometimes in disease, that's enough to have a drug that will mess up the disease. If you look at here, there's different treatments for leukemia. There's a treatment for preventing transplant rejection, so like turning down the immune system when you want it to be turned down. And there's an antibiotic. A treatment for gout is down here. There's lots of different things. All of life is based on nucleotides. And so making modified nucleotides won Elion the Nobel Prize. She got 45 different patents and 25 honorary doctorates. That's a lot. Here's a picture of her. If you want to know more about her, she's a great story about women in science. You can go to this article right here. And the other thing that's important, if you remember during COVID, people were talking about the first line of defense was an antiviral we had already developed, we pulled off the shelf, and it worked well against this coronavirus, at least better than anything else we had at the time. And it was called remdesivir. Remdesivir is in the upper right corner here. And if you look back at the previous slide, it kind of looks like those. It's a modified nucleotide. And in fact, in the upper left corner, we have a normal nucleotide, ATP. And so ATP looks like that. Remdesivir kind of looks like it, but it has weirdnesses to it. In fact, the remdesivir will mess up the polymerase that is trying to use the ATP to make DNA or RNA. And that's how it works as an antiviral. There are other antivirals underneath remdesivir, and you can see that they follow the same scheme. You just try different things, and they will always have side effects because other cells need to use nucleotides. They'll accidentally grab onto one of these and have problems. But you have the case where the side effects are worth it, because if it gets rid of the virus, then that's the desired outcome. So the important thing, upper left is a normal nucleotide. Upper right is an antiviral of the sort that Gertrude Elion started to make and other people followed. What they found out is that there is an actual, same thing like this. There's an enzyme that acts like Gertrude Elion. It will take nucleotides and it will mess them up. And it will put different chemical groups on them in cases where you are infected with a virus. The virus is very hungry for nucleotides. So what you do is to say, oh, you want some nucleotides? Here, I'll give you some nice nucleotides. And the virus will be activated to make weird nucleotides that will be toxic. It will mess up the virus. So previous papers had already found this viperin enzyme and the product that it made, the antiviral nucleotide that it made. Okay? What this paper actually does is it actually looks for breakdown products of that result in urine so that we can test urine to see if it has a lot of this antiviral thing in it. That will tell us the patient has a lot of viperin going on, and they found out that this does correlate with COVID infection. There's a lot of viperin, and therefore there's a lot of the modified nucleotide in the system. It gets excreted as a breakdown product in urine. So, and the thing about this is if you look at the center top, there's an NMR magnet up there. They use a lot of NMR and they use a lot of mass spec, and that's the biochemistry that they used. The other thing is if you look in the middle, you have NMR signals, and they found certain NMR signals that when you looked at healthy people, you got no signal. You see that. If you look at infected people, you see a signal. And they even were able to get structures from that, and the structures were that these were modified nucleotides. Okay, this is what was known before. This is what viperin will do. It will take CTP, normal nucleotide on the left, and it will convert it to a dideoxy dehydro form of the CTP. This is messed up. And if you know the biochemistry of how DNA is made by joining the 3' hydroxyl to the 5' hydroxyl. If you look on the right side and look for the 3' hydroxyl, you can see it on the left side. It's labeled right there. But it is missing. There's no 3' hydroxyl. Any polymerase that takes up this DDH-CTP molecule will be basically terminated. Its chain will be terminated, and it will stop making DNA or RNA. In fact, this is RNA because it has a 2' hydroxyl. And so viperin makes a lot of this DDH-CTP molecule. The other biochemical note, notice that it uses this molecule called SAM, which is a cool cofactor that we talk about just a little bit in biochemistry. It's important here. So even the things we talk about just a little bit in biochemistry, you end up seeing them in other places. Okay. So, and the other important thing is viperin is part of an inflammatory response. It's an antiviral response. When T helper cells see a lot of interferon alpha 2, they will make viperin. When macrophages see interferon gamma, they will make viperin. So different cell types will respond to different interferons, but the idea is the same. And we'll have an answer at the end for why different cells might have different responses to different interferons. So what this does, this is their figure of showing what happens. They're looking at T helper cells, so they say when interferon alpha 2 comes in on the left side, it will make viperin, and you follow the arrows. The viperin will take normal CTP, and it will change it to DDHC-TP. That DDHC-TP will block the RNA polymerase. Look at the little RNA polymerase thing. There's a little block sign to it. Now, the thing is, there's a bunch of this CTP going around. DDH-CTP is, some of it will block the RNA polymerase, and some of it will be left over. When it's left over, everything in your body, phosphates are very precious to your body. So it's not just going to let this DDH-CTP float around with its phosphates. It will actually have a nucleotidase that will actually break off the TPs. And so you'll end up with DDHC. That is molecule 1, the breakdown product of what viperin makes. And that's what they already knew was there. They started this study to say, I wonder, can I find this DDHC molecule? Can I find it in urine? And then it'll be an easy way to look at things. DDHC is shown as number 1 in the bottom corner. I think I circle it. It's shown right here as the breakdown product of the antiviral nucleotide that viperin makes. And then it's shown right here, this is the actual structure And you can look at it and confirm, oh, there's no 3' hydroxyl there. This is exactly what was shown on the previous slide. So what they did is they used NMR. They used two-dimensional NMR, and they looked in the urine of COVID-infected patients, and they found it, so that's good. And they found it, it's the red peaks at the very bottom there. Look at the bottom there, and you see those red peaks. Now, the way that they purified it out, it also has very similar molecules are around it. So when they purified out the red peaks, they also found out that there were two other sets of peaks that looked very similar to the red peaks, but they were just different enough. So they used their NMR. NMR can tell you structure as well as quantity. And so, in fact, the structure is better than quantity. So they found molecule 2. Molecule 3 is sort of overlapping with molecule 1, and there's molecule 4 over there. So they're like, oh, cool. Let's look at these 3 molecules. Let's figure out their structures, and let's figure out the biochemistry that made these structures. Okay. So there's 1, 2, 3, 4 is shown on this. And what they found is when they did their special kind of NMR technique, this is one that shows you which protons are near which other protons. And there's a very distinctive set of proton signals that comes. There's a yellow signal. It's shown by the interaction shown with the yellow arrow all the way on the left. So look all the way on the left. See the yellow arrow? That goes with the yellow signals on the graph. There's a green arrow that goes with the green signals, and a blue, the blue proton goes with the blue signal at the bottom. And so they found all those signals in peak 1 and in the peaks for peak 1. If you look up and down, you see yellow, green, and blue peaks. And they also found other ones. I didn't circle them, but you can look to the side and you can see other cases where you see yellow, green, and blue peaks. Slightly moved around because these are slightly different structures, but similar enough to show up with this technique right next to. It's like going to the library and you look for one book, and you see right next to it there's a similar book. And that's actually a good way to find books, by the way. It's why libraries are important. This is like going to the molecular library and you're looking for molecule 1, and you find all these other molecules as well. This other way of looking for them is actually more sensitive, and it turned up 4 more molecules 5, 6, 7, and 8. And you see those up there. They're showing their signals, but we can just trust them on that, okay? They used NMR to figure out the structures, because you can do that fairly easily. And then you have, these are the different structures, and you can see they're all related, but they have different things tacked on, different things taken off. These are mostly, they look like breakdown products of the type that you see in the urine. Everything's been sort of beaten up and broken down. And so they worked out the biochemistry, and I just have to show you a little bit of the biochemistry they worked out. So they have 1, 2, 3, 4, 5, 6, 7, 8, and they have the arrows connecting them. And so you see that they figured out, for example, that S-adenosylmethionine played a role in making the original molecule. It also plays a role in making some of the derivative breakdown products. They also have a lot of, and they also know, for example, hydrolysis will turn a C nucleotide into a U nucleotide. And they detected the U nucleotide, and so they deduced that hydrolysis must be turning number 1 into number 3. Very cool, and they figured out all those things. Look at the bottom, though. There's a lot of question marks down there at the bottom. They know that this thing is converted, and they know what the reaction is, but they haven't identified the enzyme yet. That's enough for another paper, I'm sure. So using the NMR, it's actually easier to find structures with NMR than it is to quantify the amounts of something, but you can do it. And so they did the work to quantify the amounts of molecules 1, 2, and 3. And they show a little bit about how they go from the NMR peak to figure out how much of each of these molecules is in the blood. These are the ones that you had more of each of them. And so you see that they went for the ones that had a better signal in the first place. You don't want to go for the weak signal ones when you're trying to quantify it. They were realistic. And what they did is, look at the bottom here. They did a chart where they detected a number 1, 2, and 3, and they also detected different cytokines. And so you see here you have IL-6, you have TNF-alpha, you have IL-1 beta. None of those are red colored. Red colored means you have a strong correlation. But remember that those don't necessarily induce viperin anyways. Remember what we said. Viperin is induced by interferon gamma in macrophages and interferon alpha-2 in helper T cells. So if you look at this, well, interferon gamma, you're not getting much of a red signal there. But interferon alpha-2, it's red. Molecules 1, 2, and 3 correlate with high levels of interferon alpha-2. So that's how they concluded which part of the immune system is making the viperin in this case. It's a pretty clear conclusion that it's probably the T helper cells that are making it and not the macrophages in response to the interferons. This is why different cytokines are helpful, because if you know which cytokines go with which cells, you can detect the cytokine and say it must be with that cell. So cytokines induce viperin expression that sabotages your own nucleotides because it's worth it to get rid of the virus, and that messes up the virus because it's hungry for nucleotides. And just remember the take-home message of this: the leftover versions of these can be detected in the urine, especially these three kinds. And they are. They also found, by the way, that these are always present at low levels in healthy people. But when you're infected by SARS-CoV-2, the signals go up by 10 times or more. So always remember that you're not detecting the triphosphorylated active form of the molecule in the urine. You're detecting a dephosphorylated and then breakdown products as well. Urine's really complex because things have been broken down. And you're looking for the breakdown products, and then you reason backwards to what the active molecule was. That's an important biochemical lesson for what it is like to do biomarker type research. This looks like a promising biomarker. It's pretty exciting, actually. And so there's one last thing I want to mention. As I was researching this, I found a case where viperin is bad, okay? In macrophages, in some cases, when you're infected with Mycobacterium tuberculosis. So this is, viperin produces an antiviral. And it's present in macrophages, but when you have this particular bacterium, the tuberculosis bacterium, it will activate the macrophages, and it will start a signaling cascade. And again, the details aren't important, but look over at the beginning and end of that cascade. You have a toll-like receptor that's activated by the bacterium. You have a phosphorylation signaling cascade in the middle there. And then you have NF-kappa B. Our friend NF-kappa B is going to the nucleus, and it's making these genes that will produce more cytokines in an innate response, even nitric oxide production, that will inhibit the tuberculosis bacterium survival. So just notice how all these things are coming together. But viperin can inhibit that signal. It looks like the immune system does this deliberately, because it's like, look, I can only take care of one infection at a time. So if there's viperin activated, I'm going to be trying to take care of viruses, and that will turn down the bacterial signaling. So if you're already viral infected, and then a bacterium comes in, your immune system deliberately ignores the bacterium, because the viperin is busy enough, and the macrophage needs to be antiviral, not antibacterial. It's important to see this and to see that there are trade-offs in immunity. You can't have perfect immunity. You can't fight off multiple infections at one time at no cost. It's all a cost, it's all a trade-off. And you do have a trade-off between antiviral viperin and macrophage NF-kappa B signaling inflammation in this case. And that's part of what you need to know about immunology. All right, thanks for listening this week. It was 19 minutes, not 15, so it's close enough, within 20%. And I will see you. Enjoy your break, your time off, and I will see you on Wednesday to continue this story.